Okay, let's dive into this. Welcome, everyone, to today's Explainer. We're going to take a pretty fascinating journey today through the fundamental principles of medical toxicology. And we are going to do this by taking a micro to macro approach, meaning we'll start way down at the fundamental chemistry, looking at how toxins actually operate on a cellular level, and then we'll build our way all the way up to see how those tiny microscopic reactions literally dictate the life-saving clinical treatments used in emergency rooms every single day. Because, if you want to really understand how to reverse a toxicological emergency, you have to start with the molecules.
So our roadmap for this explainer directly mirrors the source material. We're going to quickly hit 1. The chemistry of toxins, 2. Metabolism and cellular injury, 3. Neurotransmitters in the balance, 4. The physiology of withdrawal, and 5. Connecting all these principles right back to practice.
All right, starting with Section 1, the chemistry of toxins.
To really wrap our heads around toxicological emergencies, we have to adopt a precise structural perspective. The actual physical shape and properties of atoms and compounds dictate exactly how they're going to interact with our physiology. So, let's anchor this entire explainer by defining one absolutely crucial word: xenobiotic. A xenobiotic is simply a compound that is foreign to a living system. And these foreign invaders injure us in two main ways, either by totally disrupting our critical metabolic processes or by causing direct physical structural damage to our cells. Understanding how these foreign compounds behave, how they get absorbed, distributed, and ultimately how they attack is really step 1 in medical toxicology.
Take a look at the creation of that toxic intermediate right there, NAPQI. This is structural damage in action. When reactive oxygen species alter lipid membranes, they cause total membrane destruction through this cascading lipid peroxidative effect. But luckily, our bodies have built-in defenses. We have these antioxidant systems like glutathione, acting as sort of a trap for these reactive species. Usually, glutamate and glutathione protect the liver cells, the hepatocytes, from the toxic metabolites of super common drugs like acetaminophen. But when those natural defenses get overwhelmed, those highly reactive xenobiotics bind directly to the hepatocyte proteins, and that causes massive liver injury.
Moving right along to Section 2, metabolism and cellular injury. Okay, so the body uses this two-step biotransformation process to try and get rid of toxins. Phase 1 basically adds functional groups to lipophilic or fat-soluble xenobiotics. Weirdly enough, this actually converts them into more chemically reactive metabolites at first. Then Phase 2 steps in to conjugate or bind those reactive products with other molecules to make them water-soluble. Now, you might be wondering, why does this matter? Well, it's absolutely crucial because nonpolar lipophilic toxins cannot be easily peed out by the kidneys. This whole two-phase conversion to water solubility is the only way the body can expel these dangerous xenobiotics. If this process gets disrupted, well, the toxins just accumulate.
But the thing is, toxins are incredibly deceptive. Sometimes a xenobiotic wears a disguise, right? It gets mistakenly picked up as a natural substrate by our own enzymes. For example, the rhodendicide fluoroacetate is so deadly because it cleverly hijacks our own cellular tricarboxylic acid cycle. Other times, the metabolism happens in one place, but the injury strikes somewhere else entirely. In this super bizarre twist of chemistry, cyanide can actually be formed by the liver metabolizing certain nail polish removers. But that resulting cyanide acts systemically, completely shutting down cellular respiration everywhere else.
That brings us to Section 3, neurotransmitters in the balance.
If you really want to understand the brain's baseline homeostasis, you just have to look at this giant physiological seesaw. On one side, we have our excitatory neurotransmitters, primarily glutamate, which is the main excitatory driver in the central nervous system. And then on the other side, we have our inhibitory neurotransmitters, like GABA and adenosine, which work to lessen oxygen requirements and sort of hyperpolarize the neurons, keep them calm. When a xenobiotic enters the system, it tips this balance. Too much excitation, and the patient goes into seizures or agitated delirium. Too much inhibition, and you're looking at central nervous system depression, coma, and respiratory failure. Looking at the physiological synthesis and release pathways for a really critical neuromodulator, dopamine. Dopamine is fascinating because it's so incredibly concentrated in the basal ganglia and the limbic system. When xenobiotics cause excessive dopaminergic activity, it can produce severe paranoia, psychosis, or drive that really intense craving we see in addiction. But conversely, if a toxin or a drug drops that dopaminergic tone, it causes severe extrapyramidal disorders, think acute dystonias and Parkinsonism. Which leads us to this absolutely brilliant quote from the source material. All action in human neurophysiology is disinhibition. Really think about that for a second. In our central nervous system, excitatory neurons want to be firing constantly, but the inhibitory neurons hold them back. To create any physical action, to literally do anything, the brain's natural baseline inhibitory tone has to diminish. Action only happens when we take the brakes off.
But here is the pivotal question. If our brain adapts its brakes to deal with the constant presence of a depressive toxin, what happens when that constant toxin is suddenly removed? And that is Section 4, the physiology of withdrawal. When a patient takes a depressant chronically, their brain fights back to restore that seesaw balance. And it does this through these surprisingly rapid neuroadaptations. It basically pulls levers across 6 main mechanisms. It alters gene expression via mRNA. It tweaks second messenger effects via cyclic AMP. It literally physically pulls receptors inside the cell via endocytosis. It shifts where receptors are located in the synapse, changes intracellular signaling, and modulates neurosteroids. Essentially, withdrawal is a state of massively reduced inhibitory transmission. The brain has downregulated its own natural brakes, so when the drug is removed, the patient is suddenly thrown into a highly dangerous state of totally uninhibited autonomic excitability.
We absolutely have to draw a hard line here and distinguish withdrawal from tolerance. Withdrawal has 2 really strict requirements: a pre-existing physiologic adaptation to a xenobiotic and decreasing concentrations of that xenobiotic. Tolerance, on the other hand, is really just a shift in the dose-response curve, meaning, you just need more of the drug to get that same initial effect. While dependent patients almost always have tolerance, tolerance itself does not require the continuous presence of the drug to prevent withdrawal.
So why does all this dense molecular minutia actually matter in the ER? Let's do a crucial check-in with a specific example, alprazolam, which is commonly known as Xanax. Alprazolam is a high-potency triazolobenzodiazepine. Because of its specific molecular triazyl moiety, chronic use causes highly unique conformational changes to the GABA-A receptor. Because of this structural change, if a patient goes into alprazolam withdrawal, standard ER treatments like diazepam often just won't work. The receptors have physically changed so much they don't even recognize standard benzos anymore. To save the patient, doctors often have to actually re-administer alprazolam itself and then initiate a really long, slow taper.
Finally, Section 5, Connecting Principles to Practice.
And this brilliantly illustrates exactly how this micro-level biochemistry dictates macro-level life-saving clinical decisions. Consider the comparison between 2 totally deadly toxic alcohols, methanol and ethylene glycol. Now, both are actually relatively non-toxic by themselves, but they follow zero-order metabolism via the enzyme alcohol dehydrogenase. It is their acid metabolites that kill. Methanol is metabolized into formic acid, which leads directly to severe retinal injury and irreversible blindness. Ethylene glycol gets metabolized into oxalic acid, which literally causes crystalline precipitation within the renal tubules, just destroying the kidneys. But because we understand the specific metabolic pathway, we know exactly how to treat it. We administer a drug called fomipizole. Fomipizole is a competitive inhibitor of alcohol dehydrogenase. By blocking that specific enzyme, we stop the creation of the toxic acids entirely, giving the body time to safely excrete the parent alcohols. It's an elegant chemical solution to a chemical problem.
So we've traveled all the way from the atomic structure of a xenobiotic through the hepatic enzymes, across the synaptic seesaw, and straight into the intensive care unit, which leaves us with this final thought. If addiction, tolerance, and overdose are driven by such rigid, predictable, and fundamentally chemical neuroadaptations, how could truly understanding these molecular principles change the way our society views, stigmatizes, and ultimately treats addiction and overdose? It's not a moral failing, right? It is structural chemistry. Thank you so much for joining me on this explainer. Keep asking questions, and I will catch you next time.